Okay, figure 1 shows a type of gene mutation. This one is normal DNA. Here is normal DNA. This one is abnormal DNA. So here you look at amino acids here. Leucines is going to be same. Threonines also same. Proline also same. Okay, glutamic acids, now it changed to valine acid here. So why glutamic acids is changed to valine acids, valines? Because of here, thiamine is replaced by adenine. So since thiamine is replaced by adenine, so it will encode from glutamic acid to valine acids. So that we call as base substitution. Okay, what is meant by base substitution? Okay, base substitution is when one or few base pair in the nucleotide sequence in the genes is replaced with another base pair. Okay, I repeat. What is meant by base substitution? One or few base pair in the nucleotide sequence in the genes is replaced with another base pair. Okay, so state the type of gene mutation. Okay, type of gene mutation is base substitution. Okay, so we have 4 type of gene mutation. The first one is base substitution. The second one is base insertion. The third one is base deletion. And the last one is base inversion. Okay, base substitution, one or few base pair in the nucleotide sequence in the genes is replaced with another base pair. Okay, what is meant by base inversion? Okay, base inversion occur when 2 base pair or more are inverted in the nucleotide sequence of a gene. Okay, 2 base pair or more are inverted in the nucleotide sequence of gene. Okay, what is meant by base insertion? Base insertion, you add one or few base pair in the nucleotide sequence in a genes. Okay, what is meant by base deletion? Okay, base deletion is loss one or few base pair in the nucleotide sequence of a gene. Okay, so name the genetic disorder by the gene mutation as shown in figure 1. So this one, when thiamine is replaced by adenine, so glutamic acid, okay, it will produce valine acid. Okay, so this one we call as sickle genetic disorder, sickle cell anemia. Okay, remember, sickle cell anemia, it's happen when glutamic acids replaced to valine acid. Why glutamic acid replaced to valine acid? Because thiamine is replaced by adenine. Okay, describe the symptoms of the affected person. Okay, sickle cell anemia, symptom is, first is physical weakness. Okay, second is insufficient supply of oxygen. And the third one is organ damage. I repeat again, what is the symptom of sickle cell anemia? Physical weakness, insufficient supply of oxygen, organ damage. Okay, list 2 other type of genetics mutation. So we have base substitution, so base inversion. What is meant by base inversion? 2 base pair or more are inverted in the nucleotide sequence of the genes. Okay, base insertion. Base insertion is add one of few base pair in the gene, in the nucleotide sequence in a gene. Base deletion. Base deletion, loss one or few base pair in the nucleotide sequence of a gene. Figure 2 shows the normal sequence of an mRNA and its corresponding translates amino acids. So mRNA is transcribed from DNA templates, or DNA. So meaning that for mRNA it must be complementary with DNA. So if here 5 prime, DNA will be 3 prime, A will be T, U will be A, G, C, C, T, 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 G, T, T, C, T, A, G, T, A. So this one is the DNA sequence from mRNA is for mRNA givens. So now 3 base here, 3 base, 3 base, 3 base, 3 base we call as codon. Codon is a triplet base, and one codon it will encode for amino acid, for example methionine, glutamic acid, lysine. So this table show different type of codon will encode for different type of amino acids. Okay, now the question, what is the sequence of the normal template DNA strand? So here 3 prime, TAC, CTT, TTT, GTT, CTA, GTA, 5 prime. Okay, frequent exposure of the normal gene to radiation can lead to mutation, thus resulting in the production of a mutant mRNA. What is the amino acid sequence if uracil in the 5th codon is replaced by cytosine? Okay, 5th codon, 1, 2, 3, 4, 5. So 5th uracil here replaced by cytosine. So GAC. So GAC encode for aspartic acids. It's going to be same, aspartic acids. So the amino acid sequence is guanine in the 2nd codon is deleted. Here the answers. Guanine in the 2nd codon is deleted. Here is guanine is deleted. So meaning that the new sequence is AUG, A here, from here, AAC, AGA, UCA, and U. So this one is methionine acids. This one is AAA is lysine, AAC. AAC is asparagines. AGA is AGA. Okay, here no for AGA. Here okay, cannot. UCA pun cannot. So the answer is methionine, lysine, and acetins. Number 3, figure 4 shows type of gene mutation. Normal here, gene mutation A, gene mutation B. As you know, we have 4 type of gene mutation: base substitution, base insertion, base deletion, and base inversion. So now name the type of gene mutation A and B. Okay, A, look here, guanine is replaced by adenine. So A is referred to base substitution. Okay, gene mutation B, U is missing. U is missing meaning that one base is deleted or lost in the nucleotide sequence of a gene. So B is referred to base deletion. Base deletion. Okay, now between gene mutation A and B, which is more lethal? Give your reason. So base deletion and base insertion, okay, both type of gene mutation, it can cause, we call as frame shift mutation. Okay, frame shift mutation. So what is mean by frame shift mutation? Here, what is the significance of frame shift mutation? It can cause the gene to be read in the wrong 3 base. Wrong 3 base that we call as codon. So look here, what is meant by cause the gene to be read in the wrong 3 base. So if look here, U is missing. So starting from here, change to here, change to here. So meaning that the gene to be read in the wrong 3 base codon. Wrong 3 base or codon. So meaning that it can, the reading of frame will be shift. This one we call as frame. So the reading of frame will be shift totally because of base insertion or base deletion. So it will it will produce non-functional protein. It will produce non-functional proteins. Because of what? Because the entire polypeptide will be changed totally. Okay. So question number 2, between gene mutation A and B, which is more lethal? The answers is base B lah. Why? Because base deletion, it can cause frame shift mutation here. Okay. The second answer is cause the gene to be read in the wrong 3 base or codon. The reading of frame will be shift and it will produce non-functional protein. Okay. Okay, give an example of disease caused by gene mutation. Okay, you can put sickle cell anemia, or thalassemia, Figure 3 shows the result of mutation. So now here GGC TAA CGATC GTE JTA. So now for RG here. Okay, so this one is base substitution. So T 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 16. This one 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, okay, 15. So meaning that add one more base. So this one is base insertion. Okay, this one U. Okay, remove one base pair. This one is base deletion. Okay, this one is base insertion. This one is base deletion. This one is base substitution. This one is difference here. Here from here. ATC CTAA. This one is inverse. Okay, base inversion. Base inversion. Okay, question number 1, A1, state the class of mutation. So, class of mutation, we have two class of mutation. Either gene mutation or chromosomal mutation. And of course, this one is referred to gene mutation. Okay, classes of mutation. Okay, so name the type of mutation, label R to U. Okay, R is base substitution. Okay, T is base insertion. S is base inversion. U is base deletion. Okay, give one factors that cause spontaneous mutation. Okay, there are two type of mutation. Okay, so the first one is either spontaneous or induced. Okay, for spontaneous, okay, one factors for spontaneous, either mistake during DNA replication. Okay, mistake during DNA replication. That's the answer. Mistakes during DNA replication. For induce, you can put any mutation that caused by mutagen. Any mutation caused by mutagen. And we have two type of mutagen: physical agent or chemical agent. For physical agent, example is ultraviolet ray, ionized radiation, gamma ray, alpha particle. Or chemical agents, mustard gas, nitrous acids, base analogy, colchicine or atium bromide. So, how does sickle cell anemia occur? How does sickle cell anemia occur? So, sickle cell anemia happen when amino acid valine is replace glutamic acid in a protein. So why sickle cell anemia occur? Because amino acid valine, it will replaces glutamic acids in a protein. So sickle cell anemia symptom is insufficient supply of oxygen to organ, physical weakness and organ damage. Okay, state one effect of sickle cell anemia to the blood vessel. So sickle cell anemia, sickle cell, it will clumps together. So once sickle cell clump together, it will blocking the small blood vessel and causing painful and damaging problem. So the answer is sickle cell anemia, first it will clamp together. Once it clamp together, it will blocking the small blood vessel. So step 1 effect of sickle cell anemia to the blood cells. Sickle cell anemia, first it will clamp together. After that, it will blocking the small blood vessel. Okay, what is the symptom of the disease? Like I said, the first one is physical weakness. Okay, the second one is insufficient supply of oxygen, and the last one is organ damage. Okay, now figure 8 shows the DNA base sequence for normal hemoglobin and sickle cell hemoglobin. Okay, here, as you know, sickle cell hemoglobin or sickle cell anemia, thiamine is replaced by adenine. Okay, from glutamic acid, it will replace by valine acid. Okay, define gene mutation. Okay, what is mean by gene mutation? As you know, we have 4 types of gene mutation: base insertion, base deletion, base substitution, and base inversion. So what is mean by gene mutation? A change in the nucleotide sequence, change in the nucleotide sequence, okay, in a gene. Okay, a change in the nucleotide sequence in a genes. Okay, identify the class of gene mutation shown in figure 8. So, figure 8 is sickle cell anemia. Thiamine is replaced by adenine. Okay, from glutamic acid replaced by valine acid. So, this one is base substitution. State 2 type of mutagen. As you know, 2 type of mutagen. The first one is physical agent. The second one is chemical agent. Okay, example for physical agent is UV, ultraviolet ray, gamma ray, or alpha particle. Chemical agent, mustard gas, okay, chlorine or atium bromide. Okay, what will be the consequence of mutation in figure 8 in the affected person? So, what is the consequence of in figure 8 in an affected person? So, it will be physical weakness, okay, organ damage, or insufficient supply of oxygen. So, state 2 classes of gene mutation that could cause frame shift mutation. There are 2 types of gene mutation that can cause frame shift mutation. There is base insertion and base deletion. Okay, next, base substitution mutation introduce a stop codon in DNA base sequence of hemoglobin gene. What kind of mutation shown by these conditions and predicts its effects? Okay, so for base substitution, there are three types. Okay, there are three types. The first one is missense mutation. Missense mutation. The second one is nonsense mutation. The last one is silent mutation. Okay, so for missense mutation, okay, it will alter codon, and once it's altered codon, it will produce different amino acid, like here, sickle cell anemia, that we call as missense mutations. It belong to base substitution, and we call as missense mutation. Why? Because it will alter codon. Here, codon will be changed from CTT to CAT. Codon, remember, codon is 3 base. Okay. So missense mutation, it will alter codon, change to different amino acid. This one we call as missense mutation. Okay, nonsense mutation, it will alter codon, but it will alter codon, it leads to stop codon. Okay, we call as UAG, UAA, UGA. One, it alter codon to stop codon, that we call as nonsense mutation. So nonsense mutation, it lead to produce non-functional protein. It lead to produce non-functional protein. Okay, you call as nonsense mutation. So silent mutation, the last one, it will alter codon, but that codon, it will produce the same amino acid like before. Okay, it's silent mutation, alter codon, but it will produce for the same type of amino acid. Okay, this question, base substitution mutation, is introduced by stop codon here. Stop codon, UAA, UGA, UAA, UAG, UGA. Okay, we call as nonsense mutation. Okay, what kind of mutation? The answer is nonsense mutation. Okay, what is effects? It will produce non-functional protein. In this question, non-functional protein of hemoglobin. So once it's produced non-functional protein of hemoglobin, okay, it will, the hemoglobin will not function improperly or not at all. Question number 6. Figure 10 shows the result of gene mutation with their original base sequence. Okay, here, base sequence, okay, mutant A, here change, this one is base inversion. Here CA, okay, 1 2 3 4 5 6 7 8 9 10. 1 2 3 4 5 6 7 8 9. So meaning that at one base, meaning that we call as base insertion. Okay, next is GCA 1 2 3 4 5 6 7 8 9 GCA AT G. Okay, here thiamine is replaced by guanine. So this one is base substitution. So what is gene mutation? Gene mutation is a change in the sequence of nucleotides bases in a single gene. Okay, what is meant by gene mutation? A change in the sequence of a nucleotide bases in a single gene. Okay, name type of gene mutation produce mutant sequence in figure 10. So 3 marks here. Mutant 1 is referred to base inversion. Okay, mutant 2 is referred to base insertion. Okay, mutant 3 is referred to base substitution. Okay, give one example of genetic disorder for mutant 3. So, as you know, sickle cell anemia, or you can write down the thalassemia, sickle cell anemia or thalassemia. Which mutant sequence can cause frameshift mutation? Okay, this one is of course mutant 2. Why mutant 2 can cause frameshift mutation? Okay, because of it can cause a shift in the reading frame. A shift in the reading frames. Okay, state other class of gene mutation that can also cause frameshift mutation. So there are two types. The first one is base insertion and base deletion. Okay, state type of mutagen which cause mutation. So mutagen, we have two types. The first one is physical agent. The second one is chemical agents. 7. Figure 11 shows the result of two altered sequence. So what is mean by mutation as shown in figure 11? So now we have here original sequence, original base sequence. This one is altered sequence and altered sequence N. So now for altered sequence M, what you see that here thiamine is replaced by guanine. So this one is referred to base substitution. Okay, here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. This one is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 14. So this one is base deletion. Okay, both of base substitution and base deletions is belong to gene mutation. So in A, what is mean by mutation as shown in figure 11 is what is mean by gene mutation. Okay, gene mutation as usual is a change in the sequence of nucleotide bases in a single gene. So classify the mutation shown in figure 11. So mutation M is referred to. Okay, classify the mutation. Okay, this one is referred to. Classify the mutation shown in figure 11 is gene mutation. This one is meaning of gene mutation. Name mutation M and N. Okay, M is referred to base substitution. N is referred to base deletion. Okay. No name to other mutation than 3C1. So in gene mutation, we have 4 types. The first one is base substitution, base deletion, base insertion, and base deletion. So 2 other type is base insertion and base inversion. Okay, which sequence in 3C1 has more serious effects? Of course, base deletion. And give reason for your answers in 3C1 because it can causes a shift in the reading frame. And another one, different polypeptide produce, and of course it will form non-functional protein. Okay, give one chemical substance that cause gene mutation. This one is chemical agent, so mutagen belong to chemical agent. So either you put mustard gas, okay, nitrous acid, cholecycline, or ethidium bromide. All these answer will be correct. Question number 8. Figure 12 shows a classification of gene mutation. So we have sickle cell anemia for X, base insertion, base deletion, and this one is we call as base inversion. Sickle cell anemia, X refer to base substitution. Okay, define mutation. What is mutation? Mutation is permanent changes in the amount, arrangement, and structure of DNA. Okay, I repeat again, permanent changes in the amount, arrangement, and structure of DNA can be passed to next generation. So the key point here, if you want to define the mutation, is permanent changes in the amount, arrangement, and structure of DNA. Okay. So identify the type of gene mutation X and Y. X is referred to base substitution because it causes sickle cell anemia when glutamic acid is replaced by valine. Okay, Y is referred to base inversion. Okay, step 1, class of gene mutation that may cause frameshift mutation. So either you put base deletion or base insertion. Both is correct because both can cause frameshift mutation. Okay, now why frameshift mutation is said to be harmful? Because it causes a shift in reading frame. When cause shift in the reading frame, what happen is changes in codon So when it changes in codon, it will changes the amino acid sequence. You have to know that if the change make from codon is a triplet base, so meaning that codon alter codon, so it will alter the amino acid sequence. So at the last, different polypeptide are produced. I repeat again. Why frameshift mutation is said to be harmful? Because the gene to be reached, the reading of gene will be shift. So meaning that the change in codon, it will change the amino acid sequence. So meaning that it will produce different polypeptide are produced, and of course it will form non-functional protein. Okay, which type of gene mutation cause sickle cell anemia? Okay, this one is base substitution. Okay, give one difference between nonsense and silent mutation. One difference between nonsense and silent mutation. Both are belong to base substitution. Both are belong to base substitution. Okay, what is meant by nonsense? Nonsense, it will alter codon to stop codon, while silent alter codon but produce same amino acid. So as you know, we have three type of base substitution. The first one is nonsense mutation. Nonsense mutation, it will alter codon to the stop codon. Stop codon is referred to UAA, UGA, and UAG. Okay, and produce non-functional protein for nonsense mutation. For silent mutation, it alter codon but produce the same amino acid. Okay, next question. Among symptoms of sickle cell anemia are fatigue, pale, and shortness of breath. Explain why. Because sickle cell, it will block blood flow to the organ. Sickle cell block blood flow to the organ. So in blood, low level of oxygen. Why? Because sickle cell block the blood flow to the organs. In blood, low level of oxygen. Question number 9. Figure 2.1 shows type of small scale mutation that affects mRNA and amino acid sequences. So this one we have a wild type mRNA here, mRNA. This one is silent mutation, meaning that it will alter the codon but it produces the same amino acid. Look here, same amino acid. Mutation 1, okay, look at the amino acid. So glycine is replaced by arginine. So now either you base substitution. Okay, G is replaced by adenine, and then the alter codon produce different amino acids, so we call as missense mutation. Okay, mutation 2, it will alter codon to stop codon. Remember, stop codon is UAA, UGA, UAG. So it alter codon from CAA to UAA to stop codon, that we call as nonsense mutations. Nonsense mutation. And mutation 3, look here, ALGY, this one, all produce a different amino acid. Why? Because here CCA, ACC, AGG, ACA, AGA, UGG. So meaning that the whole sequence of amino acids will be changed. So this one we call as frame shift. The reading of gene will be shift. Okay, so identify gene mutation 1, 2, 3. So mutation 1 is referred to missense mutation. Mutation 2 is referred to nonsense because it's alter codon to stop codon. Mutation 3 is frame shift. Among 1, 2, 3, which is less severe? Okay, less severe is mutation 1, missense mutation. Why? Because it's only changed one amino acid in polypeptide sequence. I repeat again, mutation 1, why missense mutation is less severe compared to nonsense and frame shift mutation? Because of it's only changed one amino acids in polypeptide sequence. Compared to nonsense mutation, it produce non-functional protein. It will produce non-functional protein, and of course the frame shift will be more severe compared to mutation 1, compared to missense and nonsense, because it will change the amino acid sequence, and of course it will produce a different polypeptide sequence, different polypeptides. Okay, so give two differences between mutation 1 and mutation 3. What is the differences between missense and frame shift mutation? The first one, for missense, mutation 1 is less severe. Mutation 3 is more severe, of course. Okay, mutation 1 is only involve one codon. Missense mutation only involve one codon. Here, one codon only. Okay, so sorry. Yeah, one codon. So, but mutation 3, frame shift mutation, involve many codon. Many codons. Okay, here we have 1, 2, 3, 4, 5 codon. Question number 10. Figure 6 shows mutation at a codon in a gene coding for beta globin. So this one is normal hemoglobin, this one is mutant hemoglobin. So now what is the differences is thiamine, original's base sequence, is replaced by adenine. Okay, thiamine is replaced by adenine. So you know that this will cause the sickle cell anemia. Okay, beta globins. Okay, now name the and define the gene mutation shown above. So this one is base substitutions. Okay, what is meant by base substitutions? Replacement of one pair of nucleotide, one or few base pair of nucleotide with another pair of nucleotides. Okay, base substitution, replacement of one pair of nucleotide with another pair of nucleotide. What is the effects of gene mutation in figure 6? What is the effects of gene mutation in figure 6? It will cause sickle cell anemia, in which glutamic acids is replaced with valine acids. So it will cause sickle cell anemia. Step 1, effects of mutant beta globin. So mutant beta globin, it will cause red blood cell become sickle shape. Okay, so causing red blood cell reduce, so have reduced ability to carry oxygen.